Hello. In this video, I'd like to share with you some thoughts and ideas about how to come up with better chemical research projects and about the quality of chemical research. Young professors and students often struggle with finding interesting areas of research and may lack time to consider the implications carefully. I've studied and worked at 6 different universities and at 2 companies in 5 different countries and had ample opportunity to see how academic and industrial researchers think and how they develop their projects. Let's start with some basic economics. This chart shows the annual percentage of income spent on necessities like clothing, food, and housing in the United States from 1901 to 2023. The green line is the total expenditure. We see that this percentage has declined over the years because of the falling inflation-adjusted prices of clothing and food, while housing has remained more or less constant at 25%. These falling percentages represent rising prosperity. What is prosperity? Well, the less we spend on necessities, the more we have left for unnecessary expenditures, like entertainment or luxury. We also have to work less and thus have more spare time for family life and hobbies. To work less, the average value generation per worked hour must rise, and prosperity is the result of rising productivity. With the widespread adoption of new artificial intelligence tools, our productivity, and hence our affluence, is likely to rise even faster than before. Advances in productivity, however, require an active effort and will only arise under competitive pressure. The more ruthless the competition, the faster productivity and prosperity grow. Competition from China, for instance, is a fantastic opportunity for Western societies to improve their own productivity and thereby increase their wealth. Because competition is unpleasant, nobody likes it, and people avoid it by all means. To increase prosperity, countries need strong, politically independent institutions that protect a free market and private property from greedy politicians. I've listed here some of the main anti-competitive practices which are responsible for poverty or below-average economic growth. In autocracies, and also in many democracies, there is no or only little competition among civil servants, which leads to inefficient public services, corruption, and an ever-growing public sector at the expense of the private sector. In countries like Switzerland and the United States, there is strong tax competition between cantons and states to attract companies and people. And in both countries, the public administration is efficient and inexpensive. Other forms of competition avoidance include price fixing, barriers to entry to markets, and lobbyism. I could spend hours listing examples of such behavior, but I'll stop here. Enough has been said and written about these issues. At the end of my video about job opportunities for chemists, there is a list with some books on the subject, and in his book, The Alpha Strategy, Pugsley accurately describes how these anti-competitive strategies work. If we compare countries with each other, we see that the more money people have to spend, the lower the percentage spent on food. The poorest countries are autocracies, and countries that chose communism or socialism as a form of government. After the Bolshevik Revolution in 1917, the Russians quickly found out that communism doesn't work, but they didn't have the strength to acknowledge it. In the 1950s and 60s, when most former colonies regained independence, communism still appeared economically viable. Because their leaders viewed capitalism not as freedom, but as the cause of imperialism, and encouraged by Russia, China, and Western left-wing academics, many former colonies adopted socialistic policies. Deep poverty, weak institutions, and rampant corruption and environmental destruction were the result. Why all this talk about the economics of affluence? Because organic chemistry contributes a great deal to our wealth and quality of life. Thanks to modern agrochemicals, such as fungicides, herbicides, insecticides, and fertilizers, farmers have become more productive. Food prices have fallen, and the quality of food has risen a lot. With the aid of modern food preservatives and hygienic plastic packaging, the shelf life of food has increased. Plastic packaging is lighter than glass or metals and saves a lot of energy during the transport of food. And the incidence of food poisoning and food contamination by fungi or bacteria has fallen, which is one of the reasons for falling rates of gastric cancer and other forms of cancer. Mycotoxins produced by fungi evolved over millions of years to cause cancer and to kill animals. And although proteins are quickly degraded in our digestive tract, some bacteria produce toxic proteins like botulinum toxin, tetanus toxin, cholera toxin, or anthrax toxin, of which the lethal oral dose can be less than 1/10 of 1 milligram. No man-made synthetic chemical comes close to the toxicity of natural products. Life and evolution is just the all-out war between nucleic acids, everyone fighting against everyone with ever more deadly weapons for nucleic acid building blocks to make copies of themselves. Although decried for their high prices, modern drugs reduce the number of sick days and hospitalizations, and thus the overall medical expenses. And new materials made possible many technological improvements, which led to higher productivity. Clothes are today much cheaper and of better quality than 50 years ago, and smartphones, electric vehicles, and small portable drones have been made possible by new polymers and the new lithium-ion batteries, again, the result of many decades of chemical research. A few words about the types of scientific research. Research is often classified as basic or applied research. The goal of basic research is to satisfy our curiosity, while the ultimate goal of applied research is to make money. But projects can also be both basic and applied. For instance, a completely new chemical reaction discovered by doing basic, high-risk research, that is, research with a low probability of success, might enable a less expensive synthesis of some important compound. Such accidentally useful results are rare, however. Some sciences rest upon basic research exclusively. We study astronomy, archaeology, paleontology, history, zoology, or botany just because we want to know about the universe, our past, and about the existing animals and plants. The knowledge attained is useless and has no impact on our daily lives. Many people do not consider the satisfaction of our curiosity a worthwhile goal, or God forbid, something worth spending real money on. Scientists doing basic research therefore often try to rationalize their projects, claiming that their results might potentially have some practical use. But this is just a lack of honesty. There are also people, many German academics for instance, who despise applied research and insist that real, pure research must be basic, without even a hint of some potential application. I think that prosperity is a precious good worth fighting for, and that scientific research becomes way more interesting and stimulating if it aims at solving, directly or indirectly, some important problem. Fortunately, chemistry is a science where research can be both basic and applied at the same time. Of course, only affluent societies with strong competition and free markets can afford basic research, while poor societies, with all their equality and social justice, whatever that may mean, must do without basic research. Most industrial and academic chemical research is about the elucidation of reaction mechanisms, the discovery of new reactions, and the synthesis and study of new compounds or classes of compounds. Unlike astronomy or paleontology, chemical research can be both basic and applied. Thus, a better understanding of reaction mechanisms can help to improve a synthesis. While industrial research projects are often short-term and low-risk, academic projects can be longer-term and more risky. Let me show you some examples of basic chemical research. This here is a recent example, without any potential practical application. The authors investigated the reversibility of 4+6 cycloadditions to gain a deeper knowledge about the activation barriers, kinetics, and thermodynamics of this reaction. Today, such mechanistic studies are rare. With the National Cancer Act of 1971, it became easier in the United States to obtain funding for projects involving medicinal or biological chemistry. That is, areas where chemistry is more a tool than a subject in itself. Nevertheless, the National Science Foundation still continues to support basic, theoretical, and synthetic organic chemistry, like the study shown here. This here is another example of basic organic chemistry research, the investigation of the degenerate isomerization of a diazonium tetrafluoroborate. Such things are fun to know, but not really relevant. It's a bit like studying how many black holes there are in our galaxy. Interesting, but inconsequential for our daily lives. We chemists also enjoy trying to prepare compounds at the borderline of chemical stability. Even with our best molecular modeling programs, the stability and reactivity of compounds cannot be accurately predicted and must therefore be determined in the laboratory. Here, a preparation of secopyrimidine was attempted, unfortunately without success. Nobody has yet prepared a compound with a planar sp3 carbon atom. Basic chemical research can also deliver new reactions, like this one, which I discussed in my last video about the synthesis of alkylated arenes. Every new reaction makes a certain group of compounds more accessible, but we cannot know in advance if this will be of practical use in the future. At least it expands our toolbox and enhances our ability to prepare organic compounds. The larger the group of compounds accessible with a new reaction, the more valuable this reaction is. Industrial chemical research is mainly about identifying better drugs, agrochemicals, flavors, or materials, about developing procedures to manufacture compounds on a large scale, and about the continuous optimization of these procedures. Every manufacturing process entails a number of mechanistic uncertainties and potential improvements. Of course, the older the product, the more difficult it is to further improve the synthesis. But for new products, there is always room for improvements. For instance, it would be completely hopeless to attempt to find a better synthesis of any of the known vitamins, because these syntheses have been optimized over many years. Unless a new reaction, by chance, happens to enable an improved vitamin synthesis. But as I said, this almost never happens. Solutions to important problems are not found by chance, but by attacking the problem head-on from many different angles and perspectives, and by generating and testing as many ideas as possible. And to have a good idea, you must check many ideas. The never-ending optimization of products and manufacturing processes leads to a rising quality of all these products, to falling inflation-adjusted prices, and thus to rising prosperity. Academic chemical research can be basic or applied, or both, depending on who pays for it. In affluent countries, there are generous funding institutions willing to pay for any type of research, while in poorer countries, academics must often apply for funds from companies and will therefore have to focus on more practical chemical problems. At universities, it is possible to do more exploratory, high-risk research than in companies. Most revolutionary discoveries of the last century, however, were the result of applied, industrial, or governmental research, not of basic research. Examples include the transistor, the integrated circuit, the laser, the internet, lithium-ion batteries, and almost all new drugs. The goal of academic research is often to publish as quickly and as much as possible, irrespective of its relevance. With some articles, one really wonders why this research was done at all. I think that instead of boosting one's publication numbers, more effort should be put in asking the right questions and to increase the relevance of one's research projects. I'd like to share with you one possible way to come up with better academic research projects. There is certainly a place for basic chemical research, but I find it generally more gratifying to work towards some important goal. And high-risk applied chemical research can be as challenging and as interesting as basic research. I would suggest that instead of trying to justify one's research with some far-fetched rationalization, more relevant questions should be asked concerning some important goals. Here is one possible strategy. Between 20 and 40 new small molecule drugs and agrochemicals are launched every year. The structures of these compounds and their syntheses are easy to find, for instance, at the FDA or in patents. Their syntheses have not been optimized as thoroughly as those of older compounds and offer many opportunities for basic research and for finding a better synthesis. So, what is a better synthesis? When conducting a multi-step synthesis on a large scale, there are more limitations to what can be done than in a laboratory scale synthesis. Low or high temperature reactions or high pressure can be difficult to realize in a large reactor. And because time is money, industrial syntheses cannot afford a high dilution and must be run at the highest concentration possible. For safety reasons, the most reactive reagent should be added slowly while the reaction proceeds to avoid the accumulation of unreacted reagents. Mixing everything on a large scale and heating usually causes a thermal explosion and is just too dangerous. All catalysts, reagents, starting materials, and solvents should be as inexpensive as possible, and the waste should be easy to dispose of, for instance by burning. Reagents must of course have a certain reactivity, and there is no such thing as a non-toxic compound. Ideally, however, reagents and intermediates should be stable, perhaps even to water and air, and only slightly toxic or corrosive. Then, good syntheses are short, give high yields, and are designed to simplify the isolation and purification of products. Only distillation and crystallization are allowed, not chromatography. I've sketched here the approximate timeline of the development of a new small molecule drug. After the drug discovery phase, the new drug is patented with a compound patent, ideally together with many other compounds and patents, so that competitors cannot readily identify the development candidate. Then the process chemists take over and find a synthesis suitable for the preparation of the clinical batches of the drug and for the large-scale production. This synthesis is registered together with the drug and cannot be easily changed later because the impurity profile of the drug depends on its synthesis. If the drug passes successfully all 3 clinical phases, it will be manufactured by this synthesis established during process development. Once the drug has been launched, generic drug manufacturers will monitor its sales, and if these are satisfactory, they will start to develop a new, improved synthesis. If the drug is racemic, they may also attempt an enantioselective synthesis of the more potent enantiomer. And the day the original compound patent expires, the generics will be launched, usually at a much lower price. Hence, immediately after the launch of a new drug or agrochemical, there is room for coming up with important improvements of the synthesis, some of which may even be patentable. I think this is a great area for academic research groups to look for interesting synthetic problems, and even for more fundamental chemical questions. Here are some examples of recently approved new agrochemicals and drugs. These compounds are maybe structurally simpler than some natural products, but require multi-step syntheses suitable for ton-scale production. A fascinating challenge for sure. Just as an example, let's have a closer look at the reported synthesis of suzetrigine, a new analgesic launched by Vertex Pharmaceuticals in 2025. The compound is sold as 50 milligram tablets and must therefore be manufactured on a multi-ton scale. Such syntheses are usually conducted batch-wise in up to 10 cubic meter reactors. Some companies patent their compounds and syntheses as late as possible to increase the profitability of the drug, but this is dangerous. Because companies use the same commercial compound collections for screening, 2 companies may come up with the same drug independently. Only the one who patented first will get the patent. In the case of suzetrigine, Vertex decided to patent a manufacturing process in 2024, close to the launch of the drug. One starting material of this synthesis is 3,4-difluoro-2-methoxyaniline. Diazotization reaction with phenyl acetate and hydrolysis yielded an aldehyde that was then oxidized to an aryl acetic acid. Cyclocondensation with a racemic hydroxyketone gave a lactone, which was hydrogenated, reduced, and then converted into a cyanotetrahydrofuran. Hydrolysis and separation of enantiomers with the aid of a chiral amine gave an enantiomerically pure acid that was condensed with an aminopyridine and then converted into the final product by treatment with ammonia. The overall yield for these 8 steps was only 9%. Moreover, 3 expensive starting materials were required: an aniline, a fluorinated hydroxyketone, and a picolinic acid. The aniline can be prepared from trichlorobenzene by nitration, Halex reaction with fluoride, selective aromatic nucleophilic substitution, and hydrogenation. The aminopyridine was prepared from picoline by oxidation, nitration, catalytic hydrogenation, and esterification. The main reasons for the low yield of suzetrigine are the separation of enantiomers and the linearity of the synthesis. By linear, I mean that the molecule was constructed stepwise, functional group by functional group. A more convergent approach, where 2 large molecular fragments are combined to an advanced intermediate, would probably give a higher overall yield. One such convergent approach could be to prepare the tetrahydrofuran by a 1,3 dipolar cycloaddition. The required dipole could be generated by reaction of trifluoroacetone with ethyl diazoacetate or by heating an epoxide. This research project would be risky because the cycloaddition would have to proceed with the correct regio and diastereo selectivity, which is unlikely. But maybe catalysts can be found that control these selectivities, or that even mediate an enantioselective cycloaddition. Thus, a lot of new chemistry of high general utility could be developed with the ultimate meaningful goal of improving the synthesis of suzetrigine. Another chemistry worth testing is the addition of trifluoromethyl radicals to substituted furans. Similar reactions have been reported, and maybe the right stereochemistry can be attained somehow. In recent years, fluoroalkylations have been thoroughly investigated, and I've made a few videos on the subject. Another potential research project could be the selective monodecarboxylation of 4-aminopyridine 2,6-dicarboxylic acid, the so-called chelidamic acid. Some heterocyclic dicarboxylic acids can be decarboxylated just once, and because chelidamic acid is a chelating ligand, maybe the decarboxylation can be controlled by transition metal cations. Often the reported syntheses of new drugs or agrochemicals are quite good, but this should not discourage you from attempting something bold. With these new target molecules, your chances are high to find a meaningful improvement or some new relevant chemistry. So, what kind of research projects are best for chemistry students for their master or PhD work? The goal should be to get practical, hands-on experience with as many different types of reaction as possible, not to produce articles. Chemists are employed to solve difficult chemical problems, and the more reactions a chemist knows and the better his intuition for chemical stability and reactivity, the better problem solver he'll be. Thus, instead of spending 2 years optimizing one reaction, students learn more by developing a multi-step synthesis of some challenging target molecule, for instance, a complex natural product, or one of the new drugs or agrochemicals discussed above. This should ideally also include larger-scale reactions, for instance, 100 grams or so, and purifications by recrystallization and distillation. Unfortunately, in many laboratories, every reaction is poured onto a silica gel column, and the products are isolated by chromatography. Most yields are determined by concentrating a solution and weighing the residue, but such yields are irreproducible and usually too high because of residual solvents. Chromatography is no way of purifying compounds. In the real world, organic compounds must be crystallized or distilled, and students should learn to do that, also with new compounds. Crystallizing and recrystallizing new compounds is an art, and gives a realistic feel for purity and yield. Particularly instructive are recrystallizations until melting point stability is reached. For students interested in medicinal chemistry, I would recommend reading about case studies, for instance in the Journal of Medicinal Chemistry, and also in the excellent book Drug Prototypes and Their Exploitation by Walter Sneeder. Detailed case studies of process development can be found in the journal Organic Process Research and Development. And then, last but not least, many different projects teach more than one big project. I learned most each time I moved to a new research group with new projects, new colleagues, and a new way to think about chemistry. So, to summarize, I find chemical projects more interesting when they aim at solving some important, real problem. Solving such problems benefits everyone and makes our society more affluent. Newly launched small molecule agrochemicals and drugs offer many opportunities for new, exciting research projects. I hope you found this useful, or at least thought-provoking. Thanks for watching, and see you all next time.